So, hello and welcome to my first attempt at the screencast-o-matic version of a lecture. Now, you may hear background noises like a dog chomping on a biscuit or a snoring cat or toenails wandering through the room, so just deal with that. Anyway, this is the last chapter for the semester, and it is on the senses. You can see I'm starting off pretty fine scale here with different types of sensory receptor. The list is pretty obvious, but chemoreceptors pick up chemicals, and thermoreceptors detect temperature, not just your skin feeling, a hot plate, but also your body temperature via your hypothalamus. Photoreceptors are only found in your eyes. Mechanoreceptors respond to physical changes to the actual cell shape. So when you scratch your arm because you got a mosquito bite, you're altering the plasma membrane on that cell, and you're able to detect that you're scratching your skin. Mechanoreceptors are also responsible for hearing, which is a weird thing. Your sense of hearing is due to vibration of the tympanic membrane, which ultimately gets picked up by mechanoreceptors. And then baroreceptors are pressure receptors. The last one here are nociceptors, which are pain receptors, and they detect chemical, physical, and pain caused by temperature. So this is going to be like tissue damage or pressure. So if somebody squeezes your arm really too hard or something and it hurts, that's due to pressure. If you get a burn, that's going to be a chemical, actually, believe it or not. That's a chemical signal because your cells are going to be damaged. You can also describe in the next section here the sensory receptors based on where they get their stimulus from. So exteroreceptors are from the outside, and there's some examples. Interoceptors are from within. So if you've got a full stomach or a full bladder or a cramp, that's an interoceptor that has detected that. And then proprioceptors are specialized interoceptors that tell you where your body position is. So not just like if you close your eyes and you can tell which way your arm is facing, which way your arm is aiming, or where your legs are. You don't have to look at them. But this is also what you use if you're doing a dive off the diving board, or a flip, or you crouch, or you do a roll or something like that. That's all going to be, proprioceptors going to have a lot to contribute to that knowledge of where you are. So I'm talking about general senses first, and then we'll get into some of the cooler stuff. Tactile, touch sensation can vary in its intensity, as you can see. So if you feel a little itch if you feel your hair move on your arm, that's actually really important information for you, because we have evolved tactile sensors that go along with our hair, because we're really prone to getting bit by insects, and insects often carry a lot of pathogens. But you can feel texture and pressure. All these things are tactile general senses. Now, olfaction or smell is interesting. This is chemoreceptors are responsible for this, and if you look at this picture, let me see if I can't draw here. So let's just see. Oops, come on, draw, it says. This thing doesn't draw? What the hell? I'm trying to draw. I'm pausing it. Alright, I think it's working. Let's see. Oh, there it is. Okay. So this right here, this area down here is where the air is going to be inhaled, so it's going to be breathed in, and it's going to pass across this mucous membrane. Those odorants, is what they call them, dissolve into that mucus and make contact with these little olfactory, they call them olfactory cilia. I'm not going to name that, but I'm not going to have you remember that, but those little ends there of those cells are chemosensitive. And if you remember when I talked about different shapes of nerve cells, of neurons, I mentioned bipolar cells, and I said they were only found in your smell and sight. Well, here they are right here. So this is a bipolar neuron right there. Those neurons extend up through these olfactory foramina. Those are those holes in the ethmoid bone that you saw when we studied the bones in lab. At least I think we got to them, but you've seen pictures. So these neurons go up here into this olfactory bulb, and this is this big yellow thing right here is the olfactory bulb of cranial nerve number 1, right? The olfactory nerve. From here, it's picked up by some other neurons that take the signal to the olfactory region of your brain. So, interestingly, these odorants, these things that you might have down here, these little deals. Whoa, what in the world's going on here? Oh no! How do I go back? No. Jeez. Let me go to the next screen. Oh, bother. Wish I knew what I was doing. Oh, you piece of junk. Okay, great. This, not again, but add on to it. In any case, those odor molecules get onto that mucus and are picked up, and then the pattern that the neurons squirt, basically their neurotransmitter in your brain, tells you what you're smelling. So garlic causes one set of neurons to send impulses to your brain. You know, other spices may send another one. Body odor may send a different pattern. So your brain interprets the smells via the neurons that send impulses to it. Now, you don't have to know all of these words, okay? I'm not going to show you an image on the test. This is just FYI. I'm covering all the stuff verbally here, hopefully. All right, let's move to taste or gustation. So your gustation is taste, and believe it or not, odorants cause odor, and tastants cause taste. So tastant is a real word. Chemicals, again, dissolve. They have to dissolve in the saliva, so they dissolve in your mouth and will signal little taste buds. And taste buds are located in and around these little papillae right here. So you see these things? These are called papillae. You don't have to know the different types or which ones are shaped which way, but taste buds get kind of, they're down here in these little nooks and crannies like this, right? So the little tastants, so whatever you're eating, gets dissolved here, and then it's going to get down into these little nooks, and it's going to make contact with these chemosensitive cells. And those cells, again, send a signal, much like the olfactory neurons sent a signal, except for they send this to the gustatory region of your brain, which then tells you that you're tasting this or that. There are 5 known tastes, as you can see: sweet, salty, sour, bitter, and umami. We know what the first ones all mean, and probably have a good idea about the second one. I mean the fourth one. The fifth one, umami, which is savory. You may have also noticed that if you have a cold or you hold your nose, stuff doesn't taste as good or doesn't taste as much, because you really use your sense of smell at the same time as you use your sense of taste. Let's move on to vision. Now we're getting interesting here. So there's the eye, and I've got a list of terms over there on the right that you should, memorize. But let's kind of describe how light works. So I'm going to get my pen here again, I hope. Yep. So if there's going to be a person, let's say that this eyeball is looking at a person, right? So there they are. The light waves bounce off of this guy, right? So there's the sun's up here, right? Light waves bounce off of this object and are going to pass. They're going to pass, in all different directions. But if you're looking at them, they're going to pass into your eye. So these light waves have to go past the cornea, which is this big shield here in front, into the anterior chamber. So everything in front of the iris, right in here, is called the anterior chamber. Now, the anterior chamber is filled with something called aqueous humor, a watery liquid. Then you get to the colored portion of the eye, which is the iris, right there. And it's in section here, so you don't see it all the way across. But the iris is kind of like the drapes, and it's got a circular hole in the middle called the pupil. So the pupil's the hole in the iris. Now, everything behind the iris is the posterior chamber. It's not labeled here, but that's this area right here. This is all posterior chamber. And that vitreous body or vitreous humor is the clear liquid that's found in there. Notice that the light passed through a lens, which is right here, And that lens is going to take that light and diffract it. And it actually diffracts it kind of like this. So the image that's projected on the back of your eye is going to be that guy, except for he's upside down. Right? So I don't know if you can see that, but there's an upside down guy back there. So the image is upside down and backwards by the time it's shown to the back of your eye, but that doesn't matter. Your brain doesn't have its own set of eyes over here looking at the rear projection screen and trying to figure out what's right side up and what's right side down, right? Let's go to the next slide here, which is a close-up of the retina. So light hits the back of the eye, which is, light hits the back of the eye, which is the retina. Got to get my pen back. I think I got my pen. Yep, okay. So light hits the back of the eye right here, this retina, this back layer, the inside layer. And one thing you'll notice is that there's a lot of stuff before you get to the parts that are photoreceptive. The photoreceptive bits are down here. These are your rods and cones. Rod cells detect function in low light pretty well, and they are responsible for sort of crisp edges on objects in your vision. And then the cones are the ones that are colored green, red, and blue here. They're not really green, red, and blue, but they do detect color. So cone, color, CO, and CO, cone, and color. But light has to go all the way through a bunch of stuff, right? It has to go through these fibers, these neuron fibers right here. It's got to go through cell bodies, multiple layers of cell bodies. It has to go through blood vessels, right? So our eye is not really built that well, right? Our eye is the product of evolution. Just so happens that our ancestors had an eye that had the sensory bits kind of facing deeper into a tissue layer, which then we took from there and evolved into the vertebrate eye. So our eyes are very similar to all other vertebrates, including, fish and snakes and other mammals and so on. One thing you'll notice is that there's this sort of region right here, which is what's called the optic disc. And so the optic disc is this area right yonder, which is where all of those neuron axons from all around the eye over here plunge back through the back of the eye. So this optic disc is also called the blind spot. And I'll show you how to do a blind spot test here in a minute. Alright, so here it is. I'm going to see if I can't show you that. So what I want you to do is, I'm going to draw a, I'm not going to draw anything. Alright, I'm going to draw a dot. No, I'm not, again. Alright, pretty sure I got it this time. So I'm going to draw a dot. There it is. And a plus. Doesn't really matter, I could have drawn two dots. But I want you to line up, I want you to close one eye. So close, let's say, your left eye. And I want you to look at the dot with your right eye. And kind of be back a little bit from the screen. But looking at the dot, don't stray off of it. Stare at the dot the whole time and bring your head closer to the screen. Now in your periphery, the peripheral vision, you can see the plus. But as you get closer to that screen, you're going to pass through a place where the plus disappears. You can look over at it if you want, and it appears, but look back at the dot and it's gone. As you move further in, that plus will reappear. Now, what's happening is that that plus, when it disappears, moves, that image, the projection of that moves across the blind spot, that optic disc in your eye, which then prevents it from being seen. Now, what you'll notice is that when you go through that blind spot, that plus turns white. So it's not like you're peeling it off the page. It's just that your brain fills in the difference. It fills in whatever's missing. So when you're looking around anytime during the day, you're missing some spots, one spot in each eye, which are filled in and not noticed by you most of the time, unless you're doing this little blind spot test. what I'm going to try here is I'm going to try to make a drawing for you to explain how binocular vision works. So on the left, I'm going to try to draw a person, a head anyway. So there's a head, and this person's going to be, you're going to be looking down on this person from above. So there's their nose right there, right? Now this is a kind of a how far each eye can see. So I've kind of got the field of view from that left eye, and then the field of view from the right eye. So we've got not quite 180 degrees vision, but a pretty wide range there in front of us. But what I want you to notice is that this spot right here, this stuff in here is seen by both eyes, right? This stuff out here is only one eye. Come on, this pen is not that sensitive. Yeah. One eye. That's not yeah. You guys get the idea. So this middle bit out here is where both eyes can, oh no. What is that? Well, as you can see, my head got erased, so I'm going to draw the head in again. Eyes, nose, field of view. And then this part right here is in stereo. You've got two different eyes looking at one object. So if I draw a little bird in here, there's my little bird. It's not really, doesn't look like a bird too much, but whatever. It's a bird, trust me. This eye is looking at it from a slightly different perspective than this eye is, right? So you're getting two different images. If you can follow along with this little activity, see if you can do this. Put your thumb up in front of your face, like hold your arm out at arm's length, and find an object in the room and block that object off for one eye. So open up your right eye and put your thumb in front of that object. Now, keeping your thumb there, switch eyes. And you'll notice that you can see the thumb, or see the object now. Switching back to the other eye, the object disappears, and so on. Conversely, you could open both eyes and hold the thumb out. If you're focused on the object in the background, you can see two thumbs in front of you and one object. If you focus on your thumbs, you see one thumb and two of whatever that object is in back. So this is how we can tell distance, how far away things are from us, at what rate they're getting closer. And this type of eye, this type of setup right here, where you've got this pretty wide range of binocular vision, is found in predators and arboreal creatures. Our ancestors were arboreal, and now we're pretty big predators. So this gives us the ability to tell how far away stuff is. I'm going to draw another little animal here. Let's see if we can see what this is. So there it is. See if you can guess what it is before I finish. See it? It's a bunny rabbit. Oh, stop that. Whiskers. Alright, whatever. Well, here's his eyes, right? They're on the side. So bunny rabbits can see in front of them. They don't just look off to the side. But that, oh no, not again. I don't know what this thing's doing like that for. So bunny rabbits can see like this and like this. Do it. Oh, you piece of junk. I don't get it. Oh, for Christ's sake. Oh, we're getting entertained by my little ability to use the technology. All right, so there's the bunny rabbit's eye, field of view again. So they can see like this and like this. But they've only got this little area right here where they have binocular vision. But they can see off their sides and pretty far behind them. So this type of structure, eye arrangement, is found in like a lot of animals that are likely to get eaten, right? So if you even see among birds, if you look at a sparrow, sparrows have eyes on kind of the side of their head, but if you look at a hawk or an owl, owls and hawks have their eyes up in front. Rabbits don't have to sneak up on grass and pounce on it, so that they just have to be able to see the predators around them and hopefully avoid them after that. Okay, the bunny rabbit's gone. Now let's go to this. Now this is a visible. This is the electromagnetic spectrum, I should say. So it's not just visible light. We see that the visible parts of it are this little area right here, right? This tiny little area. So radio waves and infrared, which is heat, ultraviolet, we know that. X-rays is when you get to the doctor, right? Gamma rays turn you into the Hulk. But we only see this really narrow little range, this little narrow strip of electromagnetic radiation. You see that wavelengths over here are very long, wavelengths over here are very short. But what I want to get across here is that this electromagnetic spectrum is the same whether you're talking about radio, infrared, visible light, ultraviolet. None of them are actually bright. Now, I mean, you may say, well, the visible spectrum of light is bright. Well, it's not. The only reason that we can see the world when the lights are on is because we have sensors to pick up that sort of those wavelengths of electromagnetic radiation. I promise you that if you turn on a flashlight and put it in a box, put it in a box with a lid, and close that lid so that it's perfectly sealed from the outside, no light gets in or out. If you close that lid with the flashlight on inside of it, it is dark in that box. It's pitch black in there. The flashlight is emitting electromagnetic radiation in that little narrow range, but there's nothing in there with sensors to pick it up. So it is just electromagnetic radiation. It's not light. If you had sensors instead for radio waves instead of visible light, the room, turning on a light would still be dark to you. But if you turned on a radio transmitter, you would see those waves bouncing off of stuff. So light is only light if you can see it. If not, it's just electromagnetic waves. All right, let's move on to the ear. There's a couple of different regions: external, middle, and inner ear, and they have their own parts. So the external ear is kind of like a antenna. If you ever hear something and know it's coming from your left side, it's because that sound got to your left side slightly before it got to your right side, and your brain's able to pick up on that. Things are louder when they're in front of you because you got those auricles are shaped in a way to kind of concentrate sound into that auditory canal. So that labeled down here, that external acoustic meatus is also known as the external auditory canal. So that's this. This is where you put the Q-tip, hopefully not too deep, and at the end of that tunnel is the tympanic membrane or eardrum, and this picks up the middle ear. The middle ear has these little bones in it called ossicles, and those include the malleus, the incus and the stapes, sometimes called the hammer, the anvil, and the stirrups because of their shapes. But what they do is that sound waves, and I'm going to call them pressure waves, all right? So I see up here I say pressure waves. So pressure waves are really like waves in a medium that will. So if you splash, water, if you throw a rock in the water, you can see the waves move across. Waves move through any sort of medium like this, like air is a medium. So as those pressure waves move through the air, they start vibrating this tympanic membrane, which vibrates these little bones, which then vibrates this little thing here, right there. That's the stapes at the oval window. Now the oval window leads to this big tube, look, this thing over here, right? And I'll zoom in on that in a minute. But the two parts are this region over here is called the vestibular apparatus and semicircular canals, and then this region over here is called the cochlea. The vestibular apparatus and canals are for balance and equilibrium. The cochlea, this snail-looking thing, is for hearing. So here we are on to the next image. This is a close-up of a cochlea, and it's been simplified. You can see it doesn't have as many coils in it, but if you look at this is that malleus, incus, stapes, and they're going to shake this little oval window. So now we're going to get pressure waves being transferred into this fluid medium. That fluid is called perilymph. And as those waves move down this vestibular canal, you can see it's called the vestibular canal. The first one that it enters is vestibular canal. As the waves move down the vestibular canal, different frequencies of pressure wave are picked up by different parts of this cochlear duct. And the cochlear duct is this middle bit right here. So high frequency or high pitch noises are picked up in this area. Medium pitch ones are picked up here. Low pitch ones are picked up way down here or even around the corner. And depending on which nerve, which neurons or which mechanoreceptors are stimulated, that determines which pattern of neurotransmitter gets squirted on the auditory region of your brain, thus getting you to hear something. So think about what I'm saying here, though. Over here, here's my eardrum on the left, right? So these are just pressure waves. These aren't making noise. This isn't noise. It's just movement of air, the different differential pressures of air in that medium. I set these little things to shaking here, and then they set this fluid to shaking, and then that fluid stimulates some mechanoreceptors, and I don't know why that keeps happening. I don't know how to use this. It's clear. Back out. Oh, I and O. Great. So that pressure in that fluid stimulates those mechanoreceptors, which then send impulses down your vestibulocochlear nerve to your brain, where some neurotransmitter squirts onto your brain, and you completely imagine what that sounds like. So the noises that we hear aren't noises until we hear them. So that same thing applies as with the flashlight in the box, in that if you make noise, or if there's a noise made, but there's nobody there with receptors to hear it, it doesn't make a noise. It just creates pressure waves in that medium. Yeah, so know these parts, though. Know that this is the cochlea, and it's made up of the vestibular canal and the tympanic canal. Vestibular canal being the first tube it enters, the tympanic canal being the second, the way out, basically. So this allows pressure to dissipate. Oh, incidentally, this eustachian canal or auditory canal right here, you could also have seen it back on the previous one. That canal equalizes pressure with the atmosphere in your middle ear. So this is why when you get on an airplane, it goes up in the air, you might have to yawn in order to get it to your ear to stop hurting. All right, here we are on the last slide. So this is shows both the cochlea and the vestibular apparatus, but concentrate on this vestibular apparatus. This is this region right here, and then these guys are the semicircular canals. So they're kind of combined. With what's called static equilibrium, which is the guy on the left over here, right? He's just figures out how he's standing. You've got in these things called maculae. There it is. Maculae are located just inside this vestibule, and they have what's called, this is them down here, they have what's called an otolithic membrane on them. So otolithic membrane right there. That is oto is ear, lith is stone, so that literally means ear stones. And there are little calcium carbonate crystals embedded in this sort of gelatinous fluid with little hair cells sticking up in there. So these little hair cells are embedded in this semi, gelatinous thing with these stones in it. So when this dude bends over to pick up his hat, right? Because everybody wears a bowler hat these days. When he bends over to pick up his bowler hat, that membrane kind of slumps forward, right? It's going to like sag forward. And that's going to go dook, 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 and it's going to tweak those little hairs, and it's going to cause them to bend. And when they bend, that sends a nervous impulse to the equilibrium portion of your brain where it tells you that your head's tipped forward or sideways or backwards or whatever. Now contrast that with dynamic equilibrium on the right. So dynamic equilibrium, this guy decides to forget it. I don't want to be a hat guy. I want to be a ballet dancer. So he goes into spin, right? Well, those semicircular canals are located on the three primary axes. You got, so basically an X, a Y, and a Z axis there. So if I rotate forward, the fluid in that semicircular canal is going to slosh in backwards, basically. If I twist my head to the left, it'll slosh to the right. So it's kind of like if you set up if you imagine a fish tank, which that's supposed to be a fish bowl, and then there's a fish in it. There's a fish, trust me. Oh no, don't do it. So now it's a fish. Nope, now it's not a fish. Stupid. I'm trying to give this fish an eye. Oh yeah. Stupid fish. Eyeball. There it is. Now he's got an eye. So if I took this bowl and I spun it, if I twisted it like 180 degrees, that fish would kind of still maintain that position, right? It wouldn't spin with the bowl because the bowl is the solid thing moving around the liquid stuff inside. So when I rotate my head this way, that semicircular canal will spin, but the fluid that's inside stays stationary. And that gives you the sensation of movement and lets you know which direction you're moving and how, where kind of angular momentum is being placed on your head, on your body or head at least. So you can see this fluid down here. If the guy spins his head, the fluid's going to rush this way, and when it rushes that way, it's going to smoosh this thing over, this little copula, which is going to, again, trigger these little hair cells. It's going to trigger the zoom function again. It's not my intent to be zooming all the time, but so these little guys get tweaked over like this, right? And then that's going to tell you that you're spinning or you're tumbling or you're rolling or whatever. If you've ever done one of those bat races where you oftentimes have to drink a beer really fast and then put your head on a bat, like a baseball bat, and spin around in circles with your head facing downward, well, that's not a natural sort of motion. We don't have any evolved response to that. So what ends up happening is that you get real dizzy because your brain can't make any sense out of all of those different motions, or that kind of new and strange motion. All right, that's all I've got. Now, just what do you study? Don't study all of the figures. Don't try to learn all these words. I'm not going to give you guys images, but do if there's bullet points there, well, none of them are bullet points. If there's words that I've typed in, study that. If there's stuff that I said, so you're going to want to pause this on occasion and take notes, because if I've covered it, you should know it, right? So good luck. This is the last chapter, like I said, and I will get a quiz out for it pretty soon. Shortly after I get the presentation up on my YouTube channel.